Okay, good. All right. If you all would like to get started, you can go ahead. I'll continue letting people in.
Okay. Well, good morning or afternoon, depending on where you are. I guess it could be evening some places. Um, I'd like to thank the ARO for giving me the opportunity to speak here this morning. Um, the topic was obviously visible to you in your invitation and on the first slide that I have up. My name's Jay Rubinstein. I'm a neurotologist with a PhD in bioengineering, and my career has been focused primarily on cochlear implants, but in collaboration with my colleague Jim Phillips, we've done a fair amount of work on vestibular implants as well. I only have one disclosure, which is currently I'm a site PI for the Decibel clinical trial. That's for gene therapy for otoferlin hearing loss. So if I can get my slides to advance. A reasonable first question, given how this field is evolving, is what is a vestibular implant? And seems like a silly question, but it's been demonstrated pretty convincingly by Sharon Cushing, Tim Huller, and others that auditory input via a cochlear implant can improve balance in deaf individuals who also have bilateral vestibular loss. It's clear that the perception of sound can help stabilize people. Sharon in the Toronto group has also demonstrated that in some circumstances, a device which modulates sound input to a cochlear implant based on postural changes can further improve balance in some cases. It makes it worse in other cases. It's clearly not ready for prime time, but it's a very interesting observation. And as you'll see in several other things that I talk about today, this approach, specifically auditory approaches to improving balance, may be clinically beneficial, but that's not the same thing as restoring gaze stabilization or vestibulospinal reflex, which I would call a vestibular prosthesis. You're about to see an example of a vestibular implant that also seems to stabilize folks, but again, it's not an approach that will stabilize gaze or restore vestibulospinal reflex. And certainly in the case of sound, it's not a vestibular implant, though it is a form of balance prosthesis. There are three groups internationally that have done work with human implantation of the semicircular canal ampullae. That would be our group, Charlie Della Santina's group at Johns Hopkins, and a collaboration between the University of Maastricht and the University of Geneva. And in a very timely way, a couple of months ago, there was a review of all three group's work published, and that's the citation there. I think it was from July of 2023. I could be wrong about the month, but it's a very nice summary of the work of all three groups that have done human implants with a semicircular canal-based device. Now, another type of vestibular implant has been described by the group in Las Palmas, Spain. And this is a device that actually goes into the vestibule and ostensibly stimulates the otoliths. As you'll see in a little bit, it's not necessarily clear that it's otolithic stimulation that's occurring, but this device does seem to produce stabilization as well, very much in a way that seems similar to the auditory approaches that I talked about a minute ago. There's been extensive preclinical work done in animals that I'm not going to talk about today. I'm going to really focus my talk on human research with these devices. Excuse me. And we've done, in our team, we've done work in non-human primates. Hopkins has worked in chinchillas and non-human primates, and Mass Ioneer has done work also in primates and in other non-primate species. And this work has gone on for years, long before the first human implants were done. And the results of those studies is beyond the scope of this talk, because I would have to talk for several hours to review that as well. This talk is more manageable because vestibular implants have been placed in a small number of human subjects. And this is a moving target. I apologize if I've got any groups numbers wrong, but it's based on what's available to me in the literature. So as of last month, a publication from Hopkins says that they've implanted 12 individuals. My understanding is that most or all of these have gentamicin ototoxicity, good hearing, and symptomatic vestibular loss. And I'm about to specify the different sorts of patients that have been implanted by the different groups, because we have evidence in our group that the mechanism of vestibular loss definitely can impact outcome with a vestibular prosthesis. The European group in Maastricht and Geneva has implanted, I say, somewhere around 8, maybe more than that now, individuals who are primarily cochlear implant candidates. So they took people who were cochlear implant candidates and then put them into the vestibular lab. And if they showed evidence of bilateral vestibular loss, they were candidates for that study. Some of those people have vestibular symptoms, some of them may not, because again, they're primarily deafness patients who also have vestibular loss, contrasted with Hopkins, who are patients with good hearing and primarily have symptoms of bilateral vestibular hypofunction. Similar to Maastricht and Geneva, the group in Las Palmas has implanted 3 bilateral deafness patients who also had symptomatic vestibular loss. Our group at UW has implanted 7 people in total. The 4 patients implanted with our first generation device had uncontrolled Meniere's disease. And there are several different diagnoses in the 3 patients that I'll talk about in more detail during my talk today. We chose Meniere's disease at the time because we thought it was a good test group in that these individuals have severe disabling vertigo attacks. They have terrible hearing in the ear that we were implanting. And they were candidates for destructive surgery that would result in loss of hearing anyway. So, because one of the risks, obviously, of putting anything in the inner ear is causing profound sensory neural hearing loss, we chose 4 individuals where that wouldn't really matter because they were going to be losing their hearing anyway from surgical intervention to stop their vertigo. And the hearing loss associated with vestibular implantation is the biggest challenge this field faces. And the reason for that is, while there are plenty of people who have deafness and vestibular dysfunction, there's a lot more people who have vestibular dysfunction and good hearing. And the majority of people with bilateral vestibular loss have usable hearing. And so how to actually implant these devices and reliably preserve hearing is probably the single biggest challenge faced by this field in terms of extending this technology to more generalized clinical use. So again, what is a vestibular implant? And here's this is an illustration from that recent ARO summary that I mentioned. And for most of us, other than the Las Palmas group, this is what we envision when we think of a vestibular implant. In this case, it's those of you who've seen the different receiver stimulators from the different manufacturers. This is a Med-El receiver stimulator from a standard Med-El cochlear implant. but it's connected to a cochlear array and a semicircular canal array. And obviously, if the person had normal hearing, you wouldn't put in the cochlear array, you would just put in the semicircular canal array, the purpose of which is to provide stimulation to each of the semicircular canal ampullae. In someone who has deafness in the ear that you're implanting, which in the research setting is pretty typical, one might also place a cochlear implant at the same time. And so this is actually an image of a vestibular cochlear implant, but it has, one has the flexibility, obviously, to not put the cochlear array in place. So this is what I would call a vestibular prosthesis, because this device attempts to restore normal motion sensitivity associated with head movements. And obviously, one needs an external device that senses those head movements and sends them to the implant and encodes them in a way that the vestibular system can decode. A system that requires no such modulation was described by Angel Ramos, and he leads the Las Palmas group that I've been talking about. And they did something rather different from what the rest of the field has done. They basically took two cochlear implants and put one of them into the cochlea and the other one into the vestibule, with the idea of providing stimulation to the otolith organs. And if you look at a CT scan of their first two subjects, the left is their first subject, the right is their second subject, one can see an electrode array in the cochlea, and one can see three, you can even count them, 3 electrodes within the vestibule of that same ear, with a similar arrangement here. And they don't modulate their stimuli going into the vestibule. They provide an ongoing unmodulated pulse train. And they've demonstrated, I don't have any of their videos, but their videos are pretty remarkable in terms of the gait stabilization that this device can provide. And this is just comparing posturography results without the device and with the device active. And while balance is clearly still abnormal with the device active, it is significantly improved with the stimulation provided to the vestibule. You'll notice I didn't state stimulation provided to the otoliths because, as you'll see in a bit, it's unclear exactly what this device is stimulating. So this approach may very well prove to be extremely clinically useful, but it cannot replace vestibulo-ocular or vestibulospinal reflexes. One needs sensors that measure head movement and modulated stimuli of the vestibular end organs to restore anything approaching normal function. But having said that, these devices are simpler, and either an auditory-based approach or an approach like the Las Palmas group describes may prove to be clinically useful sooner than the other approaches I've talked about. So what conditions might one treat with a vestibular implant? The index condition is bilateral vestibular hypofunction. This is the vestibular equivalent of profound sensory neural hearing loss. And in order to restore anything looking like normal function, one needs a prosthesis that provides modulated stimuli where the modulation is based on the movement of the head. And so obviously a system like that requires some sort of head motion sensor. As I've just said, the Las Palmas approach requires no such sensor. And there is another condition, what we call poorly compensated unilateral vestibular loss, that could potentially be treated with a device like the Las Palmas device. These individuals' primary complaint is that they have chronic unsteadiness. They've typically suddenly lost vestibular function in one ear from a condition called vestibular neuronitis. And while most people who have that will compensate adequately so that they're not symptomatic, there are a small population of people with that condition who never compensate properly. And it's possible that some kind of unmodulated ongoing stimulation of the vestibular end organ may significantly reduce symptoms. And I think the results from both from Las Palmas and the auditory-based results that I've talked about provide examples of that. So either of these conditions may or may not be associated with hearing loss that's amenable to a cochlear implant. Therein lies, again, the biggest challenge, is that if the vestibular loss is not is in an ear that has normal hearing, one needs to face up to the risks of a hearing loss associated with any kind of vestibular implant. So this was our team's first attempt to develop such a device. This was our first generation implant. It was based on the Nucleus Freedom receiver stimulator and had what we call the pitchfork electrode array, where we had 3 arrays, each of which had 3 individual electrode contacts on them. And the idea behind this was that it's an extremely small array that would fit into the paralymphatic space of the semicircular canal, and ideally not obstruct the endolymphatic duct, and not obstruct the membranous semicircular canal, sorry. And the idea is we implant it adjacent to the ampulla and get all 3 electrodes close to the ampulla. And this cartoon is the idea. The sizing of this thing was 0.15 millimeters, so that it could, again, in theory, go into the bony semicircular canal without compressing the membranous semicircular canal. And the idea behind this was to minimize the risk of vestibular loss and hearing loss by minimizing inner ear trauma. The reason for the 3 electrode contacts, there were a couple of reasons for it. We wanted to be able to record compound action potentials while we were stimulating. That requires at least 1 other electrode. And we also wanted a backup electrode in case of electrode failure. Electrodes fail in cochlear implants with some regularity. Thankfully, receiver stimulator failure is much less common than that. But individual electrodes fail pretty commonly. And with these experimental arrays that are much smaller than the typical clinical cochlear implant array, we thought that the risk of electrode failure would be high. And so we wanted a backup electrode on the device. And we devised an external prosthetic, a 3 degree of freedom accelerometer that encoded head velocity on 3 audio frequencies. And Kai Bao Ni, who I earlier saw on the call, wrote some magnificent software for the cochlear speech processor that allowed us to extract those 3 audio frequencies and use them to encode modulation onto the pulse trains that the device provides. And we originally envisioned this to attach externally to a BAHA, bone-anchored hearing aid abutment, which would provide a rigid fixation of the device to the human skull. And this is a cartoon of how we might attach it to a non-human primate skull in the lab. After we did this in 10 non-human primates, the FDA approved us to implant up to 10 human subjects at UW Medical Center. They had to have what the American Academy of Otolaryngology classifies as a definite Meniere's disease. They had to have failed medical management and be a candidate for routine surgical intervention. And the idea behind it was that if the device failed to control the Meniere's disease, we could do routine vestibular surgery afterwards and take the device out if needed. So these 4 individuals were implanted between 2010 and 2012, and the outcomes of those 4 individuals are extensively described in an article that Jim Phillips published in the Journal of Neurophysiology in 2015. And from a challenges standpoint, all of them lost hearing and ipsilateral vestibular function postoperatively. This is in contrast to our non-human primates, who had some degree of hearing loss and some degree of vestibular loss, but primarily both were remarkably well preserved. One big difference between the primates and the humans is the humans had Meniere's disease. Meniere's disease is known to make the inner ear more susceptible to injury. That's been known amongst ear surgeons for a long time. Excuse me. And so from the standpoint of demonstrating preservation of hearing, our choice of Meniere's disease was poor. Obviously, for other reasons, our choice of Meniere's disease was good in terms of not hurting anybody with a first-in-human trial. And what we studied these folks over the course of 1 to 5 years, and we looked at electrically evoked eye movements and electrically evoked compound action potential, electrically evoked postural sway, and electrically evoked percepts over that time period. In terms of how we actually do the procedure, this is a surgical image from our first subject, VI01. And what I've done here is what's called blue lining the posterior semicircular canal. Now, what blue lining is, for the non-surgeons in the audience, is exposing the endosteum of the semicircular canal. And through a surgical microscope, it looks blue, or more accurately, it might be called gray lining. And you can also see that I've made a little fenestration in the endosteum with a micro pick to create a very, very small opening. And then the idea is you put the electrode arrays through those small openings. And this is the electrode array in the posterior semicircular canal. This is the electrode array in the lateral semicircular canal. And this is the electrode array in the superior semicircular canal. You can see part of the receiver stimulator back here, and then a stitch securing the electrode array into the mastoid to stabilize those electrodes. And electrode stabilization was a really important thing to achieve, because as you saw from the previous image, these electrodes are very, very short. They were 2.5 millimeters in length, and there's not a lot holding them in the superior canal. And in our monkey studies, we found, not uncommonly early on, until we figured out how to better stabilize the array, that the arrays tended to come out because the monkeys pull on their ears a lot postoperatively. To ensure that the device was activating the vestibular nerve, we recorded intraoperative compound action potentials. And these are compound action potentials from the lateral and superior semicircular canal. These are recorded using routine clinical software provided by Cochlear for cochlear implantation. And for people who are used to looking at cochlear ECAPs, these are very similar in morphology. They're just smaller in amplitude because each branch of the vestibular nerve has a much smaller number of fibers than the cochlear nerve has. This is what the electrodes look like postoperatively. Here's an electrode in the posterior semicircular canal, and the ampulla is over here. And so we're at some distance from the ampulla with that electrode. And in our animal studies, we found that the distance of the electrode from the ampulla had a big impact on how well we could activate that semicircular canal ampulla. That's not terribly surprising. So we endeavored in subsequent patients to get the posterior canal electrode much closer to the ampulla. This shows an electrode sitting literally right in the superior semicircular canal ampulla. And then here's an electrode sitting in the lateral semicircular canal ampulla. So it is possible to get electrodes into all of those locations. And then we did a variety of postoperative testing on these individuals. This is subject number 1. This is my colleague Jim Phillips over here, and we're looking at electrically evoked postural sway on the right, and here we're just evaluating his perception and his eye movements in response to electrical stimulation of the vestibular system. And he's wearing the critical piece of attire for this study, which is a T-shirt that his wife made for him, where she calls him Monkey Boy 11, since he was the first human to receive this device. The sort of 1-year summary of this first generation device was interesting, but didn't bode well for the long-term use of that specific design. We found that rotational perception, postural sway, and nystagmus are not uniformly linked. We had some subjects who would have robust canal plane rotational percepts, but no eye movements, and other folks who did have eye movements. We found that the device was unlikely to be a useful intervention for Meniere's disease, but they did help us design a better device in the second generation that did not suffer from the third bullet, which is perhaps the most important thing we found with this device. We found that while we could evoke robust eye movements early on, over time, those electrically evoked eye movements declined. And we don't have proof of precisely what was associated with that decline. But because these electrode arrays are so small and the impedance is so high because of that small surface area, we're working right at the limits of the cochlear receiver stimulator to provide current. And it wouldn't take much in the way of fibrous growth or electrode movement to basically prevent us from stimulating the vestibular system anymore. And so we suspect it's some combination of those things that led to that. We also saw some fluctuation in the electrically evoked eye movements. And we published this in 2020 in Otology and Neurotology. And what this is stimulation current on the x-axis and slow phase eye velocity from the stimulation of the lateral semicircular canal. And week 2 is in black, and we get about 10 degrees per second maximum slow phase eye velocity. And as you look over time, we see a variety of different growth functions. And if we plot the 300 microamp, 300 pulse per second stimulation over time, what you can see is there's a decrement over the first 100 weeks, but then an improvement, then another decrement, then another improvement. And these are statistically significant fluctuations in the afferent sensitivity to stimulation. One could argue that perhaps it's a change centrally, but it just didn't seem that way. But again, we can't prove that, but this looked like fluctuating afferent excitability. And if true, that would change our understanding of the pathophysiology of Meniere's disease from what we understand now. Remains to be seen if that's true. Based on those results, we devised a second generation device, and also based on the nucleus freedom implant. And again, we have the pitchfork electrode array, but we added a cochlear implant array. This is the hybrid L cochlear implant array that we added to this. This other lead is a ground electrode that was common to this device and the first generation device, and to a routine Cochlear Corporation cochlear implant. We made these electrodes 40% longer. So they are 3.5 millimeters instead of 2.5 millimeters long to provide a little more mechanical stabilization. we designed the original one to be really short so as to minimize trauma. But it was clear to us that we needed larger electrode contacts and better stabilization. And both of those things require a longer intravestibular or intracanal portion of the array. And we went from 3 contacts to 2 contacts. With 2 contacts, we can stimulate and record compound action potentials. With 2 contacts, we have a backup electrode in case one of the electrodes fails. But if one of the electrodes fails, we can stimulate, but we can't record compound action potentials from that array. That's what we trade for having larger surface areas. And we doubled the surface area of the electrode array. And we changed the clinical criteria for a candidate for this study so that they were either labyrinthectomy candidates, as they were in the first generation, or they could have symptomatic vestibular loss with speech discrimination of less than 50% in 1 year. Because the idea was that if they had less than 50% speech discrimination in 1 year, the cochlear implant electrode on average would restore that. So there would be individuals who potentially would have reduced speech discrimination in that year. There would also be individuals who got better speech discrimination with the device, but on average, the performance would be the same as it was before they underwent this experimental surgery. And as it turned out, our first subject had previously had a bone-anchored hearing aid placed in that ear, and we were excited because that would provide us, without additional surgery, it would provide us a place to mount the external sensor, or what we call the bone-anchored rotational prosthesis, or BARP. Unfortunately, for reasons you're about to see, we never got to use this. So she was 57. She had bilateral vestibular loss and unilateral deafness in the left ear. And her history was that she had sudden sensory neural hearing loss 10 years earlier, and immediately after that began having oscillopsia, symptoms of bilateral vestibular loss. And at the time, shortly after her hearing loss, she underwent a bone-anchored hearing aid placement on that side, which she was using. And she was profoundly deaf in the left ear, had normal hearing in the right ear, and had profound vestibular loss bilaterally. And so this was the first patient to receive our combined vestibular and cochlear device. And here's the electrode in the lateral semicircular canal. Here's the electrode in the posterior semicircular canal. And you can see now we're right next to the ampulla. There is a learning curve to doing this, but we did get better at it. This is the superior semicircular canal electrode right here. And this is the intracochlear electrode right here. And intraoperatively, we found no vestibular compound action potentials, none. But she had fantastic cochlear compound action potentials. We were obviously not pleased to see this because it suggested what we would ultimately find, which is she had no vestibular percepts or eye movements from stimulation of any of the vestibular electrodes. And she had normal results with her cochlear electrode. And so from a research standpoint, she was a failure. From a clinical standpoint, she was happy. She's very pleased using her cochlear implant as a single-sided deaf cochlear implant user, but not the kind of results you want to see in your first in-human experiment with your new generation device that you're all excited about. So subject number 2 we pushed on was a 75-year-old woman who had had bilateral Meniere's disease for 50 years, but it had burned out. She was inactive. She was no longer having vertigo attacks, but she did have severe unsteadiness and oscillopsia with severe to profound sensory neural hearing loss bilaterally and bilateral vestibular loss. She did meet cochlear implant criteria bilaterally and elected to undergo this procedure with a combined vestibular cochlear implant in the left ear in July of 2018. And intraoperatively, we found beautiful vestibular ECAPs and cochlear ECAPs, and she ended up getting great speech recognition with her cochlear implant. Here's images of her electrodes. This is her intracochlear electrode. This is her superior semicircular canal electrode. And here's her lateral canal electrode here and posterior canal electrode here. And this is what her eye movements looked like. So in the interest of time, we'll focus on the lateral semicircular canal, where you can see she has much bigger. The biggest response we got in our first set of subjects was a little over 10 degrees per second of eye velocity. She was able to achieve 10 times that. And so clearly the improved surface area or enlarged surface area was a big benefit for the device. And other things that you can see on these images is that while she's got rip-roaring horizontal eye movements, she's got a little bit of vertical eye movement too. More than a little bit, it's fairly substantial. That's presumably from current spread to other canals or to the otoliths. Our electrode in the anterior or superior canal, depending on whether you're a physiologist or a surgeon, showed large vertical eye movements and relatively little in the way of horizontal eye movements. These results were pretty stable over time. That is, unlike the first set of subjects, we had eye velocities that remained pretty constant over the year after surgery and beyond. So we were much happier with this device. The other really terrific finding in her was that when we put the sensor on her in a rotary chair, we could compare her VOR with and without stimulation of the device. We can restore, at least at this frequency, pretty good gain. It's actually in the normal range. It's probably not normal for her, but it's in the normal range of VOR gain when we provide modulated stimulation to her device. So that was a really terrific finding. This is 9 months out from surgery. We did a third subject, also had bilateral Meniere's disease. He'd previously had a vestibular nerve section in the left ear that was complicated by profound deafness. That was done elsewhere. And he had a left cochlear implant from Advanced Bionics in that ear. He was a successful user of his cochlear implant. He had severe to profound deafness in the right ear with bilateral vestibular loss. And also in 2018, underwent surgery with this combined device. Intraoperatively, we recorded both vestibular and cochlear ECAPs. He had a fair amount of residual hearing in that ear, which was lost. He also developed great speech discrimination with the device. And so he actually uses a cochlear device on the right ear and an Advanced Bionics device on the left ear. I actually saw him a couple of days ago clinically, and his wife wanted to know what was going on. When are we going to bring them back into the lab? He's unfortunately developed Alzheimer's disease, and he would not be the greatest subject at this point in time, but still very interested. Also had rip-roaring eye movements in response to a stimulation of his canals. Interestingly, we get sort of paradoxical responses from his vertical canals. One of the canals, the eye movements are in the wrong direction, something that we still don't quite understand. But very, very robust electrically evoked eye movements that were stable over the course of a year. With these subjects who have both a cochlear implant and a vestibular implant, we can begin to look at questions about how might one affect the other. And the effects were small. They were published in Audiology and Neurotology in 2020, but they were real. Whereas stimulation of the vestibular electrodes did not produce auditory percepts, and stimulation of the cochlear electrodes did not produce vestibular percepts, there was an interaction. And what we're demonstrating here is how stimulating the intracochlear electrodes affect eye movement responses to stimulating the vestibular electrodes. And while the effects were small, they were significant to the point where we do expect that in the future, if such devices are used clinically, this is something that's going to have to be compensated. Charlie Della Santina has demonstrated that that eye movements may not align perfectly with canal stimulation, but that through combinations of canal stimulation, it's possible to produce very specific eye movement directions. And so clinically, that's likely to be necessary in any such device. And likewise, if they have a cochlear implant at the same time, it may be necessary to attend to how auditory percepts affect the vestibular programming. These are subject number 2 and subject number 3 wearing the sensor, which is strapped onto their head, and our very kludgy power supply and processor in their little front pack while they ambulate around the lab. And what became clear with this generation device was that the internal device was stable. It produced the results that we were hoping to get, at least in 2 of the 3 subjects. And the time has come to actually build an external device that doesn't require all of this stuff to be effective. It really should look like a cochlear implant speech processor. That's a very costly project. We have not figured out quite how to fund that yet, but that would be our next problem to solve in order for our research team to move forward with this. We clearly saw in subject 1, and so here's an example of a challenge. This is a non-Meniere's patient. This individual had sudden sensory neural hearing loss with sudden vestibular loss in the same ear, who had had a pre-existing vestibular loss in a normal hearing ear on the other side. And while the cochlear implant worked great, she had literally no response from the vestibular implant. And we know it's not device related. We know the device is working normally. This was a biological problem. So one of the challenges will be how to identify diagnoses that are associated with non-responders. We don't have a good answer to that yet. Charlie Della Santina has demonstrated that at least in one patient who had had gentamicin ototoxicity for more than 10 years, I believe, had great responses. So duration of vestibular loss may or may not be related to this. There's evidence that it's not, but clearly the mechanism of the vestibular loss can have an impact. In 2 of our other subjects, our 2nd generation device appears to be able to provide much higher intensity of vestibular activation than its predecessor. In 1 of the 2 subjects, we were able to restore normal sinusoidal horizontal VOR gain, and it subjectively made her feel a lot better. That's pretty common in the Hopkins subjects as well. Subjective improvement is larger than objective findings like eye movements. And the vestibular responses to stimulation after 12 months were stable in subjects 2 and 3. Going to take just a minute to sort of talk about how etiology of vestibular loss may impact results with these devices and try to explain some of the other sort of puzzling findings in this field. So this isn't a human temporal bone, it's a guinea pig temporal bone, obviously. But I use it because it gives us a really nice image of the relationship between the cochlea, one of the ampullae, the vestibule, and the neurons and nerve fibers in the internal auditory canal. when you put in a cochlear implant, your neurons the neurons that you're targeting are in Rosenthal's canal and the medialis, and there may be variable survival of the peripheral axon depending on duration of deafness and a lot of other factors. But your neurons are very close to you. When you have an electrode in the semicircular canal ampulla, the cell bodies you're stimulating are all the way down here in the vestibular ganglion. They're far away. So if in fact vestibular neurons degenerate after deafferentation in the same way that cochlear neurons do, then trying to stimulate those neurons from that distance would be problematic. The data from the Hopkins patient conflicts with that. So again, this is one of those challenges that we really need to better understand in order to make this work. Secondly, if you put an electrode in the vestibule, ostensibly to stimulate the saccule or the utricle, you're very, very close to the internal auditory canal. And it could be that the Las Palmas device is not actually stimulating the otoliths at all. It could just be providing generalized stimulation to the vestibular nerve and providing tonic stimulation to the vestibular nerve may be what allows that device to restore stability partially in the patients who've received it. Those of us who work on ampullary devices work on the ampulla because each ampulla is unidirectional. All of the hair cells are only coded in one direction, whereas hair cells in the saccule and the utricle are coded bidirectionally, and there's no way to control which afferents you're targeting. So it's a very complex problem of how, and at least to my mind, with current technology, unsolvable problem is how to actually stimulate the utricle or the saccule in an appropriate way. But if in fact a device in the vestibule is providing tonic stimulation to the entire vestibular nerve, that makes a lot of sense to me as to why that might be clinically beneficial. This is just a cartoon of the same idea. When you have an electrode array in the cochlea, your cell bodies are not too far away. When you have arrays in the ampullae, your cell bodies can be quite a distance away. So we may find the field of study of how cochlear implants behave and how does histology change with duration of deafness it could be very, very different in the vestibular system. But again, all of that remains to be seen. I'm going to stop there and acknowledge all of the other authors of this work. Jim Phillips actually began this project when the NIH put out a request for proposals in 2004 to do animal research on vestibular implants. And he led that contract that we had from NIDCD, and that's what led to our subsequent work in humans. Kai Bao Ni designed all the software for these devices. Leo Ling did a lot of the primate and human testing, as well as Chris Phillips, Wendy Parkinson, Amy Novak. Jason Levins from Cochlear helped us with design fabrication. And this was all enormously expensive, funded by NIDCD. Cochlear defrayed the costs of the devices partially, and a large gift from a grateful patient is what really got the project off the ground and got us to the point where we could get NIH funding to get the results I talked to you about. And I'll stop there.
Thank you very much, Dr. Rubinstein, for that great presentation. If anyone in the audience has any questions, please feel free to use the raise your hand feature in Zoom, and then I can call on you one by one, or you can just post your questions in the chat and I can ask them for you. Hi, Larry, go ahead.
Thanks so much, Jay, for a wonderful summary of the UW program. As you might guess, I have a number of questions, so I'll stick to just a few. So, so the how do you actually explain the Las Palmas results? So, yeah,
You're asking me to speculate on the Las Palmas
of
results.
course.
My first response was I didn't believe it. But I've seen videos of these subjects walking with the device activated and without the device activated. And it's very clear when it's turned on, they're more stable. It's not for gaze stabilization. It's not, replicating the vestibulospinal reflex because it's passive stimulation. It's unmodulated stimulation, but it is definitely improving their gait stability. And so my speculation is it's stimulating the entire vestibular nerve and just providing some tonic activity to the central vestibular system, which is somehow or other beneficial to ambulation. Beyond that, I don't know.
So, as a vestibular physiologist, the way we explain the dynamics of the epithelial activation is largely by throwing away spontaneous discharge. A gain measurement is you're tossing away spontaneous discharge, and it seems to me that the Las Palmas results is restoring some of that tonic discharge, which actually can be of great benefit to integrating let's call it gaze stabilization inputs from a whole host of other sensory systems, including the auditory system. Again, it's as you may know, we're pursuing. We consider spontaneous discharge part of the signal, not noise per se. That brings me to the second question: is when you deliver your pulse module, I'm assuming it's pulse modulated, frequency modulated spike pulses, correct?
We've done both frequency modulated and amplitude modulated pulses.
Fair enough. But when you actually pass through in a sinusoidal head movement, does your stimulation actually go to zero, or does it actually try to provide some stimulation in essentially what would be the null head movement state?
Yeah, the latter. In
Okay.
other words, it's tuned to there's a tonic level of activity that's modulated up and down.
Yeah. So I'm wondering if you thought whether you could reproduce the Las Palmas results either by just providing some tonic level of stimulation with a patient on a platform to see if their postural stability improves.
Yeah, no, a great thought. As well as you could also sort of do what Las Palmas did, which is just sort of attempt to quantify their ambulation.
Yeah, exactly.
No, it's a great idea.
Very good. Okay, thanks so much again.
Thank you.
I think you have a couple of other questions there, John, from Brodsky.
Disconnected from my Zoom. Can everyone hear me okay now?
Yes.
Okay, great. Yeah, Niffer Brodsky, if you want to ask your question, go ahead.
This is me coming from a physical therapy background, and I'm just asking, Dr. Rubinstein, you mentioned that your participants did report that they were feeling better and that their balance and gait was more stable. What were you using to measure their gait? Did you have anything to quantify it?
No, we did not. We did not quantify gait.
Okay. That was all.
That-
Thanks.
yeah, that's an important area of work too, for obvious reasons that come back to what Larry was just talking about, and because some of these interventions may prove to be clinically useful before we actually get the whole thing understood or working. Improving gait would be a big step forward, and more than I think some of us appreciated early on in this field. Obviously, that requires quantifying it.
Thank you.
Any other questions from anyone else in the chat?
I have one more, John.
Sure, yeah. No, go
Okay.
ahead, Larry.
I don't mean to. So two questions. Jay, have you conducted the head impulse test on any of your subjects?
We've not done it formally. We've done it informally. We don't have any data on head impulse testing.
Okay. You know any reason why you should not see any response?
Um,
In other words, it's not a limitation in the bandwidth of your prosthetic, obviously.
Oh, but it's not rigidly attached, as you saw in
I
the image. It's strapped onto the head. So it's a little more problematic to measure,
Sure.
high velocity stuff than sinusoidal.
Yeah. Okay. And then finally, if I might, John, I do so a critical element of this research seems to me that it would be great to have some postmortem morphology. Have you attempted to try to collect
Yeah,
as macabre as that is?
That is another great question. During the first phase of our work, one of our subjects died unexpectedly of prostate cancer, and I had not consented them for temporal bone harvest. Obviously, at that point, consenting for temporal bone harvest became a high priority, and everybody else that received the device consented for temporal bone harvest. Thankfully, none of them have passed away as of yet.
Good. Thanks so much.
Any other questions? All right. Well, thank you so much, Dr. Rubinstein. And if there are any other questions that get posted in the chat, we'll be sure to email those to you so you can follow up.
Have a great day, everybody.